Hello and welcome to Botany 101. I'm Angela Nishimoto. Okay, so last time we went into genetics part 1 and now we're going to do genetics part 2. Okay, we're going to proceed with a dihybrid cross. Okay, we did the monohybrid cross last time. We're going to go through some more genetic terms and we're going to continue with some more genetic problems. Okay? So during a dihybrid cross, you study 2 genes, okay? These 2 genes have to be unlinked genes. That means that these have to be genes on separate chromosomes, okay? So in this dihybrid cross, di means 2, hybrid means, of course, you're crossing 2 different things, right? So it has 2 things. So this true breeding P generation differs in 2 traits, okay? So for example, we have big G, big T, crossed with little g, little g, little t, little t. That means green potted, tall, crossed with yellow potted, dwarf. Okay? So of course the F1 generation after this cross would be big G, big T, little t, okay? Because P generation, the only kind of alleles that you can get from this is a big G and a big T, and the alleles from this other parent, it would be a little g and a little t, okay? So the F1 generation, or the first filial generation, if you remember the terminology, would be this big G from one parent, this little g from the second parent, this big T from one parent, and this little t from the second parent, okay? So then what happens is that you intercross the F1, or you self it or sib it, okay? You either self the plant or you allow it to cross with a sibling. And in this, you definitely need to use a Punnett square, okay? So here it would be big G, big T, little t, crossed with big G, big T, little t, okay? So the gametes that are produced, okay, from these, from both of them, would be big G, big T, big G, little g, big T, and little g, little t. Okay, you'll see it come up on the next slide here. Okay, so don't get scared looking at this now, okay? Just kind of relax, all right? So here's a big G, big T, big G, little g, big T, and little g, little t. And same thing across this axis. So these are all the possible gamete combinations for these 2 traits, or these 2 alleles, okay, on this side and on this side. Okay, so of course you see a lot of G's and a lot of T's, but we'll kind of break this down and maybe you'll be able to see it better. But the thing is, what it comes down to is with a dihybrid cross, when you're studying 2 genes, right, each gene having 2 alleles, okay, you come out with a 9:3 to 3:1 phenotypic ratio. Remember, the phenotypic ratio is a ratio of the phenotypes of what the plant looks like, right? So you have 9 green and tall, that would be these. Anything with a big G and a big T in it would be green and tall. So that's these plants here. This is 7 plus 8 plus 9, okay? So you get 9 green and tall, then you get 3 green and dwarf. That would be something with big G and 2 little t. So here's 1, here's a second, here's a third, okay? And you'll get 3 yellow and tall, so that would be little g's and big T, little g's and at least 1 big T. These 3 would be yellow and tall, and you get 1 yellow dwarf, okay? Not only that, but there are different 3:1 ratios. You remember when you actually do monohybrid cross, you get a 3:1 phenotypic ratio? Well, in here we have different 3-to-1 phenotypic ratios. You have 12 that are green, these 9 and these 3 that are green, so 12 that are green and 4 that are yellow. Okay, so that's a 3-to-1 ratio. Okay, you have 12 that are tall, these 9 green and tall plus the 3 yellow and tall, so 12 tall. Okay, and you have 4 dwarf. Okay, so that's another 3-to-1 ratio. Okay, you get 9 green and tall and 3 green and dwarf. That's another 3-to-1 ratio. And you get a 1-3, that's yellow and tall, and 1 that's yellow and dwarf. That's the last 3-to-1 ratio. Okay, so the most important thing to remember about a dihybrid cross using Mendelian genetics is that you get a 9-to-3 to 3-to-1 phenotypic ratio. Okay? And of course, if you have any questions about this, you can always contact me, okay? Now we're going to do a back cross, okay? That's a cross between a hybrid and its parent, okay? So in pea plants, red is dominant over white, okay? So when you cross a purebred red with a purebred white, okay, the hybrid, of course, is a heterozygote, right? You have one big R, one little r, okay? So all their offspring will be red, because red is dominant, right? So what you do is you take this F1, big R, little r, and you cross it back to its homozygous recessive parent, little r, okay? And what you'll get is 1/2 red offspring and 1/2 white offspring, okay? So we'll take a look about that, okay? So you cross this heterozygote, big R, little r, with a little r, okay? So you'll get half or 2-to-2, okay, or 1-to-1, yeah? Big R, little r, to little r. So you get half of the offspring are going to be red flowered, and half of them are going to be white flowered, okay? Now, to do a test cross, okay, you're actually having, if you have the F2 of a monohybrid cross, remember we talked about the monohybrid cross, the tall times the dwarf, right? You have 2 genetically different plants actually look the same, right? You have the homozygous dominant, right? Or capital T, and the heterozygous individual, okay? Big T, little t. So they look the same, they're both tall, okay? So if you have plants that are tall, you might not be sure whether they're homozygous or whether they're heterozygous. So you can actually cross with the homozygous recessive parent, okay? So if you cross a tall plant with a dwarf plant and all their offspring are tall, you know that the tall plant is a homozygote, right? That tall plant is capital T, or big T, okay? And if you cross a tall and a dwarf, okay, and half the offspring are dwarf, just like in the back cross, you know that the tall plant is a heterozygote, okay? So I'll leave it to you to kind of calculate this and do your own Punnett square about this particular problem, okay? So if you have any questions, please contact me about this. Okay, this is not your Learning Log 2. Your Learning Log 2 was assigned last time, okay, but you should try to practice with this problem, this test cross, okay? Now in the case of incomplete dominance, okay, neither allele is completely dominant, okay? So the heterozygote, okay, or the one that is, you know, the F1 generation, will be intermediate in phenotype or what it looks like to the 2 homozygote parents, okay? So in the case of snapdragons, the P generation would be capital R for red, R1, R1 for white, okay? Capital R1 for white. Okay, all the F1 are big R1, or they're pink. So if you cross a red flowered snapdragon to a white flowered snapdragon, they're both purebreds. All their offspring will have pink flowers. Okay. Now in the case of this incomplete dominance, okay, the F2, if you take this F1 plant and you self it or you sib it, okay, you will get 1 red, RR, to 2 R1, or 2 pink, to 1 R1, R1, and white. Okay, that means that your genotypic ratio, or 1 RR to 2 RR1 to 1 R1, R1, right, that's the genotypic ratio, is the same as the phenotypic ratio, would be 1 red to 2 pink to 1 white, okay, in the next generation, in the F2 generation, okay? So the principle of dominance was demonstrated by Mendel's crosses, okay, so this is actually another case, okay, where neither one of the pair of alleles completely dominates the other, okay? So remember that in an incomplete dominance cross, the genotypic ratio and the phenotypic ratio are the same. Okay, so here's taking a look at the snapdragons cross, okay, in terms of the Punnett square. Okay, so the red parent and the white parent have pink offspring. All their offspring are pink. So don't be fooled by thinking that they only have 4 offspring. This just means that of all their offspring should be pink, okay? You might have many more than 4. Now if you cross the F1 generation, the pink by the pink, okay, see, this is showing the Punnett square, right? So you get 1 red to 2 pink to 1 white, okay? All right, to review all the things we covered in this section, okay, even including last time, okay? We talked about DNA. We talked about DNA replication. We talked about protein synthesis, molecular genetics, okay? We talked about mutations, especially translocations and inversions. We talked about ploidy, Mendel and his experiments. We did the monohybrid cross. So remember, your monohybrid cross problem will be posted on the LaLima, or should be posted by now on the LaLima. Okay, it's due 1 week from the last lecture. We went through the dihybrid cross this time. We talked a little bit about genetic terms, and we completed the genetic problems that I wanted to go through, okay? Okay, so what we're going to do is we're going to switch gear again and get into the next section, okay? I hope you like genetics. I know it's quite involved and it's kind of hair-raising in a way, but at the same time, it's very interesting. You never, well, you can sometimes predict what you're going to come up with, okay? So today, we're going to continue with plant breeding and propagation, okay? So the outline of topics, we're going to do plant breeding and plant propagation, okay? For one thing, about crop plant evolution, okay? There are approximately 250,000 species of angiosperms, okay? So there are only 6 species that provide 80% of the calories for humanity, that we get most of our calories from worldwide. These include wheat and rice and corn, potato, sweet potato, and cassava. Okay, so these 6 crops provide 80% of the calories for all of humanity. Okay. the three big grains, wheat, rice, corn, and root crops, or potato is a stem, but sweet potato and cassava are root crops. Okay, then 8 other species complete the list of major crops. Okay, so sugar cane, sugar beet, bean, soybean, barley, sorghum, coconut, and banana. Okay, so what does this tell you here? The sugar cane and sugar beet being among the 8 species that make up the list of complete list of major crops. That means that we eat a lot of sugar is what it is. Okay. Now, hunter-gatherers, we used to be hunter-gatherers, humanity, okay, became agricultural societies, okay, and plants have been cultivated, actually on purpose, planted and cultivated for about 10,000 years, okay? And during these 10,000 years, there was development of cities and what we call civilization. Remember World Civ that you had to take maybe, okay? So you had these concentrations of humans, okay, in certain cities, which brought about some bad things like drought, famine, also epidemics, okay? But also, we have domesticated plants. We've changed them to meet our needs, okay? And domesticated plants are such that the reproduction depends on human intervention, okay? For example, if you have a corn plant, okay, a whole field of corn in fact, and you abandon it, okay, it'll actually not grow any corn anymore after 1 or 2 years, okay? So the origins of agriculture include hunter-gatherers. They maybe had better lives than agriculturalists. Okay, they generally had a better diet and they've had better health. Okay, I think these are sand bushmen that you see on this picture here. They had a lower starvation rate, which might seem like unbelievable because during agricultural times, of course, we had surplus. Okay, so we needed actually agriculture to actually have the modern capitalist state. Okay, they also worked fewer hours. Okay, their work week was about 20 hours per week. Okay, so they worked much fewer hours than we do today. Okay, so humanity's domestication of plants started among the earliest places in the Near East, okay, about 10,000 years ago, in an area by western Iran on this map here. Okay. And about 1,000 to 3,000 years later, it cropped up in Asia and the New World. In Asia, I guess in the Yellow River valleys and Yangtze River valleys, and also in the New World, okay, in the Americas, especially in Central and South America. And you had African and New World farming about 4,000 years ago. Okay. So our history with agriculture isn't very long, only about 10,000 years, and of course the human species has existed much longer than that, maybe 200,000 years. So the first crops were cereal grains, okay, things like wheat, okay? And about 1,000 to 2,000 years later, you had root crops such as sweet potatoes and yams and cassava, and legumes such as peas and beans, okay? You also had the crops of vegetables and oil and fiber and fruits, okay? So then about 2,000 years ago, people started cultivating forage crops. That's for animals, okay? Crops for decoration, for like flowers and ferns and other kinds of plants that people use in floral arrangements and bouquets and lei, etc. And then drug plants too were cultivated from about 2,000 years ago. Okay, now Nikolai Ivanovich Vavilov, okay, in the 1930s, right? He actually made the first serious attempt to find out the geographic origins of domesticated plants. Okay, he started studies that led to the understanding of relationships between the cultivated plants and their wild relatives. Okay, so wild relatives and crop plants are valuable sources of genes, of course, for crop improvements. Okay, plant breeding. Okay, the systematic breeding of crop plants has been only for about 200 years. Until then, improvements were made by planting seeds from desirable plants, but the actual systematic breeding of crop plants didn't start until about 200 years ago. Okay, so plant breeding is like accelerated evolution guided by humanity rather than nature. Okay, what it does is it forms plants with desirable alleles or desirable variants of genes. Okay, so some of the traits that we want in our crop plants include improved yield, or more crop for the investment that you put into it. Disease resistance. Okay, here you have this kind of bumpy papaya here. These kind of grooved citrus fruits. These are actually papayas. Yeah. So anyway, you want improved disease resistance. Okay, so this is a disease plant and these are healthy ones. Okay. Pest resistance. And also tolerance of stress. Okay. Now with the amount of arable land dwindling, okay, we actually need to improve the amounts of food harvested per acre. We also need to produce crops that grow on low quality land. Okay. So Norman Borlaug, okay, he's known as the father of the Green Revolution, right? What he did is he actually bred high yielding varieties of wheat, and he grew so much of it and in such places such as developing nations that people think that he probably saved more people's lives than anybody else had in history with his Green Revolution. Okay, he got the Nobel Prize for Peace in 1970, right? Norman Borlaug. Okay, now breeding methods. Okay, so breeders divide crop plants into two groups. You have self-pollinating and you have cross-pollinating plants. Now self-pollinating plants, of course, can self-pollinate. Okay, another thing about them is because they their mother and their father is the same parent. Okay, the pollen parent and the pod parent is the same parent. Okay, the mother is also its father. Okay, so they tend to be homozygous, all right, and they also tend to be inbred and also adapted to and thriving on inbreeding. Okay, some plants that do self-pollination include wheat and rice and oats and barley and peas and beans, tomatoes, peppers, apricots, nectarines, peaches, and citrus. Okay? Got all that? I'm just kidding. Okay, now we have some cross-pollinating plants. They generally cannot self-pollinate, self-fertilize. Okay? They tend to be heterozygous, or mothers and fathers are different from each other. They're different plants. Okay? Those that are heterozygous include corn and rye, alfalfa, clover, many fruits, many kinds of nuts, and different kinds of vegetables. Okay, so what they do is they do outcrossing. Okay, so that means they cross between genetically different plants. What does that usually result in? That usually results in something called heterosis. Okay, heterosis means hybrid vigor. Okay, it means the plants generally are larger than their parents, they're vigorous and healthy, they're fertile, and they're high yielding, or they make lots of fruits, lots of flowers, or opposite, lots of flowers, lots of fruits, okay, lots of grains, okay? So if you self-cross-pollinated plants, that can lead to something called inbreeding depression, okay? That's actually a term in biology. What happens is that you get small plants with poor vigor, they have low reproductive capacity, low reproductive health, okay? You also sometimes get abnormal plants because of the expression of deleterious recessive alleles. That happens also in populations of humans when you have small groups of humans interbreeding over many generations, okay? So what happens is that breeders force selfing to create inbred lines with these bad alleles eliminated, okay? Then what they do is they cross these inbred lines to make hybrid seed, okay? And these have a lot of good traits. Corn is a big one, okay? Most corn in the United States is from hybrid seed, okay? So one of the illustrations of the concept of breeding methods about this, the goodness or the functionality of this technique is that in 1908, G.H. Shaw actually had 2 lines of corn, and these 2 different lines of corn would produce about 20 bushels per acre, okay? He bred these 2 lines of corn together, and the hybrid produced 80 bushels of corn per acre on average. So you had a 4-fold increase in yield, okay? So this is something that people can do when they do breeding, plant breeding, okay? Now, heirloom varieties are early varieties, and these are European and American varieties of different kinds of vegetables usually and fruits. And what they do is they allow open pollination. They don't put bags over the flowers to prevent bees or other insects from visiting them. They let whatever is going to pollinate them. And what that leads to is you have a crop that is not uniform, and you have seeds that are more variable, which can be very good because it can produce crops under different environments, okay? For example, if you have a dry year, okay, and insect infestation, and you have these open pollinated plants, you can actually end up with seeds and plants that can withstand drought. And you can also get seeds and plants that can withstand insect infestation, okay? So what that does, it contributes to genetic diversity of breeding programs, okay? So that's actually good, you know. So this is one of the reasons why sexual reproduction is so good with the mutation, with the recombination during meiosis, and etc. that we talked about before, is that through sexual reproduction you get more variation. And this variation allows individuals to survive different kinds of conditions, okay? Now, the germplasm is a sum total of the plant's genes, okay? So plant varieties of today tend to be genetically uniform, okay? They may not be good sources of new genetic variability, okay? And this homogeneity, that means that because they're not very variable, makes them vulnerable to disease. So if anybody has ever heard of the Irish potato famine that happened in the mid-1800s, okay, what happened is that the Irish people, the Irish peasant, relied only on a very few varieties, only maybe 1 or 2 or 3 different varieties of potato to sustain most of their population. And what hit was something called Phytophthora infestans. It's an oomycete that causes late blight, okay? And for several years, the Irish people lost their entire crop, and since almost the whole population depended on potatoes, you had mass starvation. You had about 1 million people dying of starvation and the attendant diseases that come along with starvation, such as cholera and typhus. About 1 million people dying. You had about 1.5 million to about 2 million people emigrating or leaving Ireland to go to other countries. So you had people coming from Ireland and going to the United States, of course, to Canada, to New Zealand, Aotearoa, and also Australia, you know. That's why you have big populations of Irish in those various places due to the Irish potato famine. Okay? So here are some of the results of the Phytophthora infestans, late blight. The plant rots. They turn black and they rot. And the tubers, remember the stems, the storage stems, they rot in the ground, rendering them inedible. Okay? Now the thing too is that in the 1970s, the U.S. corn crop had a problem. Okay? About 15% of the U.S. corn crop was wiped out by a fungus due to the genetic uniformity of corn. Okay? So what they do is they establish gene banks worldwide, okay, to meet the needs for genetic diversity in plants. So scientists regularly go on collecting trips, okay, in areas where wild relatives of crop plants live to get new blood, so to speak. Okay, now for plants that are sexually incompatible, that means that you can't breed them together because they're not closely enough related. You have transgenic methods, okay? So species boundaries no longer exist anymore. You have things like protoplast fusion, okay? Like they've done with potato and tobacco, okay? So what can happen is you can actually do protoplast fusion or gene splicing. And these can be done when it's impossible to reproduce a plant sexually with another plant when they're not compatible with each other, okay? So what happens in protoplast fusion is that generally cells of different plants are grown in liquid media, okay, with nutrients added inside. They remove the cell walls because, of course, plants have cell walls, remember from a long time ago, okay. Then the protoplasts are mixed and stimulated with electrical current or with chemicals to fuse, okay. And what these fusions are put through is they put them through tissue culture in sterile conditions. And what you end up with are somatic hybrids combining or hybridizing body cells, okay, not through sexual reproduction, okay? So potato and tobacco, you saw a little earlier. There's also this protoplast fusion that they've done with haploid clementines and diploid mandarin oranges. So that resulted in seedless and easy to peel oranges. Okay, so they've done that with citrus too. Okay, so gene slicing in transgenic plants. Okay, they can do recombinant DNA technology where you take DNA from one organism and put it in another organism. And that makes genetically modified organisms, or GMOs. Okay, so remember that DNA behaves the same way whether it's in a bacterium or in plants or in humans because the genetic code is nearly universal, right? So genes from bacteria or from humans even can be inserted into plants' genome, okay, leading to transgenic plants, right? So what they use is something called restriction enzymes. So restriction enzymes are from bacteria, okay? They're used to restrict foreign DNA because bacteria can pick up DNA from the environment that they find themselves in, okay? Bacteria then produce these enzymes that chop up foreign DNA so the foreign DNA won't become incorporated into the bacterial chromosome and take over the bacterial cell. That's the idea behind it anyway. But these restriction enzymes we use to actually cut DNA at specific sites, okay, leaving overlapping ends called sticky ends, okay? So what these sticky ends do, and this cut DNA does, is it base pairs with other DNA that is cut with the same enzyme, okay? So I know it sounds complicated, but it's kind of simple, but it is very mind-blowing in a way, okay? So you need to clone a gene, okay? So if you're having this bacterial insect resistance gene, okay, then you need to, first of all, you need to find the gene, which is a lot easier than it sounds. Because in bacterial genome, there are thousands of genes, okay? But first you need to find the gene, and cut it out of the bacterial genome with a restriction enzyme. Okay? Then you also cut a plasmid with the same restriction enzyme. A plasmid is a small circular piece of DNA that exists separately from the bacterial chromosome. Okay? It can also reproduce separately from the bacterial chromosome. Okay? Then you mix them together. Okay, you mix a gene with the plasmid. Okay? Then the sticky ends will join, making recombinant DNA. All right? Then the bacteria multiply, and what they do is they copy the recombinant plasmids over and over again. So what that is the bacteria is actually cloning the gene that you cause to be put in the plasmid, and you can end up with millions of copies of that recombinant gene. Okay? And how to actually get it into a plant cell. Okay? First of all, you need to remove the gene from where it was cloned with a restriction enzyme. Okay? And then insert it into a bacterium called Agrobacterium tumefaciens. Okay? That's a bacteria, okay, that actually causes galls in plants. So what that will do is that will infect the plant, and it will insert the DNA, okay, from the plasmids into the plant's chromosomes. It causes the plant to produce galls that kind of house and feed the bacteria. Okay? So genetic engineers have taken out the disease-causing genes. Okay, you can insert insect-resistant genes into a plasmid, then infect the plant cells with the bacterium. Okay? And the cells can be cultured to produce whole plants expressing insect resistance. It's pretty remarkable. Okay, now, when agrobacterium technique was first developed, it was not usable for monocots. So what they did, they invented the particle gun, okay, which is an alternate strategy for getting genes into monocots. Okay, what it does, it literally shoots DNA into plant tissue. Okay, what they'll have is tungsten or gold pellets coated with the DNA, and the DNA will be shot into, say, like corn cells. Okay, and then this way they made transgenic corn, there's transgenic soybeans, transgenic cotton, transgenic canola. Okay, they've imparted disease resistance, insect resistance, and herbicide resistance into plants. They've created bioreactors to create pharmaceuticals such as vaccines for hepatitis B, for rabies, for cholera, for tuberculosis, malaria, diarrhea, and for cavities even. Yeah, amazingly so. They've created biodegradable polymers. Okay. They also use some of these GMOs to sequester heavy metals or do bioremediation. We're actually cleaning up areas using living things, especially genetically engineered living things. Okay. Ornamentals have been created. They're trying to make blue carnations, blue mums, and blue roses. Also black roses as well, that's still possible. They've actually made a black orchid, but that was through conventional hybridization. Okay. Through this gene splicing, they're able to extend shelf life of cut flowers and other flowers used and other parts used for decoration, decorative effects. They've created compact plants or plants that are smaller because generally in nature, the larger the plant is, the more photosynthesis it does, the more flowers it can make, the more fruit it can make, but they can actually make more fruit on smaller plants. Okay, and they've actually made fragrance genes or restored fragrance genes to restore fragrance quality to roses and carnations, some plants of which have lost their smell. Some people say that roses don't smell as nice anymore as they used to. Okay, so there are more than 395 million acres of transgenic plants grown worldwide today. In the United States, 93% of cotton crop, 86% of the corn crop, 93% of the soybeans, and 95% of the sugar beets are GMOs. Okay? 75% of all processed foods in the United States have GMO component. Okay? So we're eating it now. Soybean, corn, cotton, canola. Okay? So these are all about transgenic plants. So we're all eating it. So we have a gene that makes it resistant to Roundup. Okay, Roundup is an herbicide. It's a broad spectrum herbicide that will kill grasses as well as broadleaves. Okay, now some pros and cons of transgenic plants. Okay, pros. For one thing, farmers use less pesticides. Okay, there have been a 21% decrease in use of insecticides on BT cotton. This is cotton that's been engineered to produce BT, or Bacillus thuringiensis, which actually kills caterpillars. Okay. Now when Roundup is used, you don't need more persistent herbicides. Okay. Farmers also have reduced tilling, which leads to less erosion. That's some pros. Now there's some cons to it too. Okay. So of course questions about GMOs need to be answered. Okay. So, like about the effects of BT, Bacillus thuringiensis, on non-target organisms, such as other insects, worms, birds that might eat caterpillars and humans farther up the food chain. Okay. So may herbicide-resistant genes move into weeds? Okay. So you have this herbicide resistance gene going to a wild population, perhaps. Okay, if that happens, you can no longer use Roundup, okay, to kill these particular wild populations of weeds. Okay. So canola is actually a hybrid right between kale and turnip. Now canola can cross with mustard weeds, wild mustard. Okay, and in Canada, they found herbicide-resistant canola. Okay, what they are, they're actually invading wheat fields, so they can't use Roundup on them anymore because they're resistant. Okay, that's actually already happened. Could there be some insects that are resistant to BT? Okay, that's actually happening too. Okay. Might also encourage farmers to abandon sustainable agriculture. I know that sustainable agriculture is a big buzzword now, but actually these big farms actually can buy out small farmers, because farmers work very hard. They really do work before the sun comes up, and they work until after the sun goes down. They have to work very hard to make their farms produce. Okay. So what they can do, what they do is they plant BT corn before they even know the corn borer is the problem. So here's a corn borer eating its way through an ear of corn. Okay. So maybe what would be more sustainable would be to plant non-transgenic corn, then monitor and apply pesticide only when it's needed. Okay. Now another con is the human health issues. Maybe people get allergic reactions from transgenic protein product. Okay, what they had done is they put a Brazil nut gene and they inserted it into soybean to increase the content of methionine, which is low in beans. Okay, and this protein caused allergic reaction to some people who ate it. Okay, so they abandoned that project. Okay. Also, maybe animal feed might be used for human food products. Okay, the famous example of Starlink corn had a gene for BT in it. Okay, and that was supposed to be only corn used only for animal feed, but it found its way into taco shells, tortilla chips, and corn dogs all over the world. Okay. Anyway, so those are some pros and cons about transgenic plants. Okay, now we turn to plant propagation. Okay. So if you have a superior plant, a plant that's high yielding, a plant that looks really beautiful, okay, a plant that produces a lot of good things, whether crops or ornamentals, you need to propagate it. Okay. That allows thousands of acres of corn, or allows thousands of orchid plants. Okay, so we have sexual reproduction that leads to seed propagation. Here's some, looks like avocado pits. Okay, so you can get hybrid varieties from crossing inbred plants. Okay, how do you do that? First of all, for corn, you detassel it. So detasseling means removing the male flowers. So this is a common summer job for people in the corn belt, for young people in the corn belt. Okay, and then you have a row of inbred plants. Okay, and that would be next to a row of other inbred plants or another type of inbred plant. Okay, so the tassels of one is removed, and what that does, it prevents it from selfing its own ear. Okay, and then pollen comes from the other parent, carried by the wind, because of course grasses are wind pollinated. Okay, so pollination happens first, which is the transfer of pollen from a male structure to a female structure. Then fertilization happens. Okay, and the mature ears, once they mature, are collected and they're sold to farmers for hybrid seeds. Okay, so the kernels are used as hybrid seeds for the next growing season. Okay. Now, inbred varieties grown from seed, okay, they're allowed to self, right? And so what you can have is going from seed, you can get uniform population. So, so all the plants are about the same size. They all produce about the same amount of fruits, like inbred green bean, because of course a bean is actually a fruit, right? Anything with a seed in it is a fruit. Okay, so then the mature seeds are harvested and stored, and they need to be viable or still alive. Okay, so viability is best maintained in cool, dry storage. Okay, for viability and vigor. Okay, so samples are removed and tested for those for viability and vigor. And green beans and lettuce and onion and pepper, they tend to last or be viable for about 1 to 2 years. And beets and tomatoes, they tend to be viable for 5 years or longer, which is can be remarkable. You need to store them under certain conditions to allow them to be viable for that long, though. Okay. Now for plant propagation, okay, the soil needs to be moist but not sodden. Remember that the plant needs water, but it also needs oxygen at the root. Okay. So celery, lettuce, onions are cool crops. They need cool soils to grow in. They won't germinate in warmer soils. Okay. Whereas tomato and bean, cucumber and sweet corn, they need warm soils, or like summer soils, so germinate in temperate areas. Okay. So when you do plant propagation, of course, weed control is important. Okay. Because what weeds can do is they can grow really fast, and they can shade out and stunt seedlings. Okay. And they compete for nutrients with the seedlings. Okay, so people need to weed or hoe, you know, with a weed with a hoe, and also with weeder sometimes, depending on how big your bed is or how big the area you're weeding is. Okay. Now the seedlings that you're growing, desirable seedlings that you're growing, will also compete with each other. Okay. So sometimes thinning is necessary for a good crop, so you can actually sow some seeds like carrots pretty near together, but you'll be thinning them out as they get larger to make sure that the roots grow big for certain ones of them. Okay. Now, asexual plant propagation, people have been doing this for centuries. Okay, cloning plants. It's easy, and you can make many plants from just one. Okay, so you use the vegetative parts. What that does, it avoids the genetic variability and unpredictability of sexual reproduction. Okay, so question. What are the vegetative parts of a flowering plant? Do you remember? Okay, so I'll leave that for you to answer, right? So you can do this through crown division. Okay, that's parts of the crown and the roots. You can actually just divide the plant. So ornamental perennials such as gingers can be divided that way. Okay. You can also take a cutting, like a leaf cutting from African violets. Okay. So with cuttings, the plant, or from vegetative propagation, plants can be propagated indefinitely. You can actually have a plant that can live forever. Unbelievable as that might seem. Okay, think of all the tea plants, you know. So here's some adventitious roots on an orchid. Okay, so from stem propagation, you can use a tea plant for adventitious roots. Okay, so what happens is that at the stem you get dedifferentiation. Okay, and what then will happen is it'll make a new meristem. Okay, then make more root growth. So most important when you take a stem cutting is you need to control water loss, because the plant doesn't have roots usually when you cut it. Okay, so you need to keep the plant moist until the roots grow. Okay, so a good way to do it is to grow it in a closed container, even like in a baggie with paper towels around it. Okay, have it in high humidity. Okay. Decrease transpiration. Okay, so commercial nurseries use mist benches. That means that they have cuttings in pots, and then you have mists that come on every 20 seconds or so, or every minute, or every 3 minutes. That keeps the places moist. Okay, so these mist benches, they can actually stimulate rooting with rooting powders, such as with auxins in it. Okay, rooting hormones, right? And they'll usually pot them in perlite or vermiculite, or have trays or perlite or vermiculite. And these are inorganic substances. They're like volcanic rock that's been puffed up. Okay, so what they do is that they'll hold water and drain well. Okay, it's kind of a hard thing to do, to hold water and drain well at the same time, but that's what's necessary to prevent the cuttings from getting diseased if they're too wet. Okay, so some plants that are asexually plant propagated include African violets, snake plant, also known as mother-in-law's tongue, begonias, coleus, and kalanchoe, kalanchoe. Okay, an advantage to these asexual plant propagations or cuttings is you get identical copies of valuable plants. Okay. A disadvantage too is that if the parent plant is diseased, okay, the propagated plants will be too, and these include diseases caused by viruses and bacteria and fungi. Okay. Now there's a method called layering, which is a modified cutting propagation technique. It allows adventitious roots before the new plant is detached. Okay. So you have tip layering in blackberries. Okay. Other plants also too with a flexible stem can be propagated this way. What they do is they bend the canes over and cover the tips with soil. Then roots form, then shoots. Okay. Some variations is that you can actually force a stem to lie horizontally and cover it with soil at intervals, or you can pile up soil or up around the stems until adventitious roots grow out. Okay. Another kind of layering is air layering, which is usually used in tropical trees and shrubs. You can get a few large plants for one plant with a rigid stem. Okay. First you wound the branch, you either gash it or girdle it. That means you peel off the bark in a strip around it. Then you might dust it with rooting powder to encourage growth of adventitious roots. Then you wrap it with sphagnum moss, dampened sphagnum moss. Okay. And then you cover it with a plastic film to provide resistance to dehydrating. Okay. You cover it with aluminum foil to prevent overheating and to reflect sunlight. And when roots grow, the layer is removed and transplanted. Okay? Okay, anyway, we're going to stop there. Okay, and we'll continue with the plant propagation next time. We'll see you then. Aloha.